So good evening, everybody. Welcome to tonight's program. I'm Brady Newbill at the Museum of Discovery and Science, and welcome to our Distinguished Speaker Series sponsored by the Save Our Seas Foundation. Tonight's guest is Dr. Kady Lyons, research scientist at the Georgia Aquarium. Before we get started with Dr. Lyons tonight, I just want to go over a few ground rules. First of all, we ask that everybody keep their microphones muted throughout the presentation just to limit any audio interference, and we can direct our undivided attention to our guests tonight. You can feel free to turn your camera on or off as you feel comfortable. And if you have any questions throughout the whole presentation, put them in the chat. We'll come around at the end and have a Q&A session. But if anything jumps into your mind as our guest is talking tonight, you can put it in the chat at any time. We'll come back around after the presentation and save some time for questions. So, you know, just be mindful of that, but you won't be interrupting anybody in the chat. So keep those questions coming. We'll come around to them at the end. Our guest tonight, as I said, is Dr. Kady Lyons, who is a research scientist at the Georgia Aquarium, who has previously studied at California State University at Long Beach, University of Calgary, and University of Santa Cruz, California. Go banana slugs. And so really excited about tonight's talk. We're going to be hearing about toxicology in shark populations and how that can be used to know more about their behaviors and ecology. So this is going to be a very fascinating thing that we don't hear a whole lot about, and some new methods of research. So without further ado, I'm going to turn it over to our special guest tonight. So please join me in welcoming Dr. Kady Lyons. Thank you for joining us.
Thank you so much. Let me just pull up presentation. We'll go ahead and get started. Right. All right. So yeah, with that introduction, thank you so much for that. So tonight's talk is going to focus on generally pollution in elasmobranchs, but in specific, I'm going to be talking to you guys about how we can actually use this as a chemical tool to understand more about these animals' physiology and ecology. So just to back up a little bit before we dig in, it's important for us to think about traditional ecology and the ways that we study how animals interact with their environment. So one of the things that ecologists are really focused on is a concept called niche differentiation. So essentially what that means is if we think about this as a coastline and these different bubbles representing different species, it's saying how different species can use different resources while still occupying the same environment. So that might be differentiating where they are in space. So if we think about a tree, for example, you might have a niche differentiation between squirrels that live at the tops of the trees or squirrels that live at the bottom of the trees. And that would be a way that they can reduce their competition for the same resources while living in the same place. So we can have other types of differentiation through space and time. So some of the ways that we study this, at least in the marine environment, but also terrestrially, is through tagging and tracking. So that's literally putting some sort of technology on the fish. So in this case, this is just a shark fin showing a satellite tag that you guys might have seen some of this used previously. So we can tag these animals and physically track along where they go or measure the depth profile that they're using. So that's one way that we can look to see what kind of habitats animals are utilizing. Other ways that we can study this is from a diet perspective. So this would be looking at stuff like gut contents. So physically, what are these animals eating? Because that's important to tell us who they're interacting with and who is going to be important into those energy links when we start thinking about food web dynamics, which is a really important basis of ecology. So other tools that we can use are now becoming, I don't say more important, but we're starting to develop other ways that we can take different approaches to look at how animals are utilizing their environment or interacting with other species. So chemical tracers are becoming a popular tool to be able to use. So for example, stable isotopes, which you may have heard before, are taking advantage of natural phenomenon in the world. So we have isotopes that have different neutrons in them, and those isotopes move predictably through the environment in ways that we can understand their ecology. So for example, nitrogen stable isotopes have been shown to be a really nice way to look at trophic linkages in terms of trophic steps. So who's eating who, and we can use that as a predictable way of saying where on the food chain would this animal lie by taking a small tissue sample and taking advantages of those chemicals. Other chemicals that have been used also are elemental analysis. And so this is taking advantage of particular geology or hydrology in an environment that is going to impart a particular signature to a place. And you can take advantage of that to understand, okay, if this animal is here, it's going to have this particular elemental signature. So an important piece of when we think about using chemicals as tracers to understand animal ecology is there has to be a few checked boxes that need to occur in order for that to be useful. So for example, these things do need to be habitat specific, and that's in order so that we can actually reliably predict or trace back where that animal was. So for example, this is again just our hypothetical coast, and let's say that, you know, that we have this random assortment of these different elements or stable isotopes that vary either from their distance from shore or depending on the primary productivity. So who's fixing the carbon in that environment? We need to have predictable ways that are going to be habitat specific, or else it's just not very useful to us. In addition, oftentimes these need to be diet driven. So why that's important is when we think about ecology, we want it to make sure that, you know, when an animal is eating another animal, it's acquiring that signature and it's incorporating that into its body. But oftentimes when we start talking about diet and start talking about that incorporation, we need to then also think about things from the physiology side, because that might influence how often that animal is going to interact with a thing. It could influence its metabolism, so how much it's eating and how quickly it's processing those nutrients might also affect how those chemical tracers are incorporated into that tissue, and then therefore the conclusions that we can try to extrapolate from that data. So for example, thinking about things like sex or age can be very important, because oftentimes animals will use their environment differently depending on where in their life cycle they actually are. So we kind of need to take all of those into account when we're thinking about how useful a chemical tracer is as we're using that as a tool. So this is bringing up another point that we'll see very quickly, is that physically separating ecology from physiology is actually quite difficult, because for example, if we're thinking about, again, a traditional ecology, right, that the movement of an animal might influence what habitat they're using, and what habitat they're using might influence who else they're interacting with in terms of who they're feeding on. Similarly, go back. Forward, I guess. Yeah, there we go. Similarly, their physiology can influence their metabolism, which is going to be influenced by age and sex, and that could influence growth and how much tissue incorporation they're doing. So really, when we think about it, these things overlap a lot more than we think about. And so when we think about trying to separate those two, it's actually really difficult. And so our chemical tracers actually incorporate aspects of both ecology and physiology. So we need to take both of those views in order to fully understand and interpret these tools. So organic contaminants are a class of anthropogenic contaminants. So essentially what it means is that these have a carbon backbone. Oftentimes they have these 6-ring structures that you can see. And like I said, they have any number of chlorines, fluorines, bromines, so your halogens that are going to be attached to these. So in particular, some of the contaminants that I study are what we call legacy contaminants, which is a bit of a misnomer because legacy implies that these no longer are a problem, but they definitely are a problem. So some of the things that that characterize these contaminants that also might check those boxes in terms of their use as a chemical tool is that they're persistent in the environment. And so from not a health perspective, obviously that's not great for it to be persistent, but from using it as a tool perspective, that is important because we don't want something that is ephemeral, right? We want something that can, again, be reliable to the place. Oftentimes, these sorts of contaminants will be site specific, and that has to do with their history of production or use in a particular area. So Southern California, where I was born and raised and did a lot of my graduate work, has the distinguished, if we can call it, honor of being one of the largest DDT hotspots in the world. So what we see in particular for a lot of animals that come from Southern California is they will have a really particular DDT signature. And we can exploit that signature to learn more about how those animals are using Southern California in particular, or if at all. And we'll talk a bit more about that later in this talk. And then importantly, they also check off that box that they are obtained from diet. So many of these contaminants bioaccumulate and biomagnify as they move through the food web. So bioaccumulation basically just means for an animal, they will build up higher concentrations of this thing over the course of their lifetime. And biomagnification, on the other hand, is where you move up trophic steps, we see a significant increase in those contaminant concentrations. So again, all of those are acquired through diet. And so that can then have us have that twofold prong process of where it's site specific and it's going to be through diet. So again, how they're interacting with other animals in a particular environment that might have a particular signature that we can tease out. So in the past, the use of organic contaminants as these ecological tracers has had limited applications. So there was a study published in 2010 on humpback whales that looked at sampling the blubber of those animals in Alaska and measuring those contaminants in those animals. And so what they were able to do, it was the reverse. So these animals are going down and feeding at different places along the coast. And because they're feeding at these different latitudes, and we know that certain chemicals, again, based on their use and production and how they've been deposited in the atmosphere and move around, we can use that to tell where those animals are coming from. So they did this on a very large scale, but it hasn't really been done on a much smaller scale in the marine environment before. In addition, this was limited in the fact that it was only done on a single species. So it was really cool to be able to see how humpback whales are distinguishing between some of their feeding and pupping grounds along the West Coast. But again, that was restricted to a single species. So if we're thinking about an application to a whole ecosystem wide, obviously you need to have other animals involved in that. And finally, the study used few chemical congeners. And what that means is that they had few contaminants that they actually were using in their models. And the reason why that is important is because we can gain more power with the addition of more contaminants, basically. So coming back to Southern California again, we wanted to use this area as a test case to see, okay, we know that there's a large historic previous use of contaminants in the area that has a particular signature. So we might be able to use this as a test case to look to see, can we apply these contaminants as tracers to learn more about the ecology of some of the animals that live here. It's utilized by multiple species and at different life stages available in Southern California. So this is setting up a scenario where we might be able to start teasing apart, are there species specific signatures that we can look for, or is the development of an animal going to matter in terms of our ability to use this tracer? So ideally, if we're thinking about wanting to tease apart species, we want something that isn't going to be significantly impacted depending on the age of the species, because obviously that would provide a problem in terms of when we want to try to separate those species out. So a couple of our study questions that we wanted to address are twofold. So one, we wanted to say, can we use these as tracers of a metric of ecology? So in a sense, can we discriminate among species that overlap in space and time that we know we catch in the same area in Southern California? And in addition, we wanted to know that would we be able to overcome the fact that we have species with different physiological characteristics, and what particular influence might this have on our ability to use these contaminants as tracers? Because not all species are exactly the same, and they're going to have nuances in their physiology that might influence either their uptake or incorporation of those contaminants. In addition, we were also interested in kind of a second part of the project to look at the influence of ontogeny. So ontogeny is just a fancy word for life stage or development. So if we think about ontogeny, that's going to span from when an animal is born to when an animal dies. So we wanted to know, how do contaminant concentrations change across the developmental life history of an animal? And kind of some sub points under that, one of our aspects that we were very interested in was the influence of this process called maternal offloading. So maternal offloading is a process whereby females will transfer inadvertently part of their accumulated contaminant load to their offspring. So it's not that moms are evil and they're trying to contaminate their babies. This is a natural process that happens as moms are investing high amounts of resources into their young. So in particular, when moms are transferring fat in particular, that is a really good vehicle to transfer these types of contaminants, because these contaminants are what we call hydrophobic. They hate water and they love fat. So if you have something like breast milk that is full of fat in it, that is a really good vehicle that mom, again, inadvertently can transfer part of her accumulated load onto the baby. And finally, we wanted to look to see if there was some age-specific differences in physiological ecology that might influence how these contaminant concentrations change across the lifespan of an animal. So our study species in question that we're using here in Southern California are three different sharks. So we have our Shortfin Mako shark, our common Thresher, and our blue shark. So just to give a little bit of background about some of the characteristics of these species, because they will become important later as we start trying to make sense of the information that we found. So Shortfin Makos make these on-offshore migrations. We oftentimes see young using more nearshore waters than large animals. So this is very typical for many elasmobranchs as well as other vertebrates, is that once you grow larger, you're able to actually use larger spaces. But in general, when we can think about the ecology of a Mako shark, we do know that they make these on and offshore migrations. So again, the reason why that's important is that a hypothesis is the closer to shore that you are, specifically in Southern California, but really closer to areas of anthropogenic impact, that might increase your chance of contaminant exposure, because where humans are, there will be contaminants. In addition, Mako sharks have a wide diet breadth, and essentially what that means is they aren't specialists for one particular prey item. So they will feed on a wide range of things, and as they grow, across their ontogeny, they will actually increase that breadth of prey items. So again, important when we're thinking about things that are accumulated through their diet. And finally, an important thing to know is these guys are what we would call endothermic. And so essentially what that means is they're able to defend a core body temperature against the ambient temperature. So essentially they're not, warm-blooded isn't quite the right word, but essentially they are able to elevate their core temperature and defend that against the surrounding water. So again, why is that important to think about? So if we think about the fact that keeping yourself warmer than ambient, that's going to cost energy. If it costs energy, it's likely that your feeding rate might need to be higher to be able to support that aspect of your physiology. Next, we have our common thresher shark. So this guy is coastally associated. Unlike the Mako shark, it does specialize on bait fishes. And that is typically thought as that they are feeding lower in the food web compared to something like a Mako shark. These guys are what we would consider regionally endothermic, and essentially these guys are able to warm their eyes and their brains, but not necessarily their whole core like a Mako shark. And finally, we have our blue sharks. So these guys we think of more as a typically offshore species. They also have a wide diet breadth, so they're going to eat a wide variety of prey items. And these what we would consider traditionally ectothermic. So they are not elevating any part of their body and defending that against the water outside of them. So what we wanted to do then is take this physiological ecology approach to look to see, do we see differences in their contaminant signatures? And then can we use that as tracers to then distinguish these shark species, despite the fact that we know that they overlap in space and time? So part of our hypothesis that we were expecting, again, thinking that, these are contaminants that we know bioaccumulate and biomagnify. So we thought trophic position will be one of the important parts, but it's going to interact again with their physiology and ecology of how close they're spending to shore. So basically, just in general, we are expecting to see across development these U-shaped curves, and we thought that these curves might differ based on species characteristics. So if we break this down just a little bit and think about what this means for their contaminant concentrations, so focusing in on that U shape, so what we were expecting is that we would have a concentration that we're going to measure in the liver. So their livers are really fatty, so it's a good place that these contaminants accumulate, and therefore a good tissue for us to look at. So maternal offloading is going to give a starting point to that liver concentration when they're young. As they grow, we are expecting that growth to happen fairly rapidly, and this might have a diluting effect and thereby lowering the concentration, because we expected them to grow faster than they were going to be able to accumulate contaminants. Finally, as they start maturing, that growth is going to slow as they stop putting their resources into growth and start diverting that into things like reproduction. So we assumed that we would start to see this uptick in these curves towards the end, or in these larger sharks, which by extension are the older sharks. So we expected to see that while they're not growing, that they're going to start bioaccumulating, and that contaminant concentration is then going to start increasing again in our older sharks. So this is balancing this basic idea of growth versus intake when we think about the liver just as a compartment that is going to expand or contract based on their physiological state and how well they're able to feed. So what we did was we sampled these 3 species over a large range of their sizes. We then took a small sample of liver and we extracted that for a suite of organic contaminants. And then we compared signatures and concentrations across them. So when we looked at the total contaminant concentration for each of our species, I want to point out that this is going to be on a log scale. So note that it's 10 times higher for every tick mark that you see. So in general, though, we saw this really nice stepwise increase in contaminants such that blue sharks, threshers, and makos, regardless of their age size, had significantly different contaminant concentrations in terms of that total load. So even if you gave us a blind sample and we analyzed it, we would be able to say, based on the concentration alone, with pretty high accuracy, what species it belonged to. Similarly, when we looked at signatures, and in that, what I mean by this is thinking about all the contaminants that we measured in those livers and comparing them against each other. So we're able to use computer models to simulate what the accuracy of assignment would be, again, based on a signature. And again, this is regardless of age class and sex. So essentially, the important part of this little chart is to think about the diagonal, because this is the ability of the model to correctly assign the species to its actual value. And so you can see there are a few instances where the model made incorrect assignments, but overall we had a 96% success rate based on the signatures to be able to distinguish our species. And so if we put that in 2-dimensional space, just so you can see visually what that would look like, and mapping out our 3 species, they have these really distinct places that they're falling, again, in 2-dimensional space, but we're looking at a wide suite of contaminants, suggesting that there is niche partitioning of some sort going on among these animals. And that's probably reducing that competition in Southern California in areas where they overlap in space and time. So this kind of checks off that first mark, that it does seem that we can use organic contaminants as tracers by 2 different means, both on total concentrations, but also the signatures in the livers. So next we wanted to look at those ontogenetic changes. So if you'll just recall, we're expecting to see a maternal offloading bump where we're going to have young animals start out with high concentrations and over their juvenile stages undergo that growth dilution and decrease in concentration with size. We expect there to be a point where they reach some sort of equilibrium. And again, that's when their intake of contaminants based on what they're eating is matching their ability to dilute that with their growth. And then finally, when we get to the adult stages, that the intake would exceed excretion, and then we would see this again climb in concentration as they age. So looking at maternal offloading, we were able to, we didn't have any moms and pups that we were able to match up, but we did have some young of the year sharks that were around the size that these sharks are born at. So we were pretty confident that the loads that we were measuring in these animals were going to be reflective of things they acquired from mom and not things that they acquired on their own when they were feeding. And we saw a very similar pattern to the one that we saw previously, where we had Makos kind of taking the cake in terms of contaminant concentrations, followed by Thresher sharks and blue sharks. So if we break all of these guys down and look kind of over their ontogeny, so over size, for our blue sharks, and again, breaking these out just based on some different classes of our organic contaminants. So as you can see, it's kind of a scatter shot across the graph. There is no significant change as these species are growing. So note, we did have mostly juvenile sharks with some of our, go back, please. mostly juvenile sharks or subadults with few adults. But nevertheless, we're not seeing those patterns that we expected to see. So we're like, okay, that's not meeting our hypothesis. Kind of a bummer, but maybe the Threshers will show something. So we looked at Threshers, we saw that decrease over size that we were expecting, but it kept decreasing. We didn't see that increase in contaminant concentration like we were expecting for any of our classic contaminants. We're like, okay, all right, we're 0 for 2, awesome. But finally, when we looked at our Makos, we actually did see that U-shaped curve for our contaminant classes. So phew, yay. In the sense that we did see these high levels in these young sharks, a decrease over size, and then a subsequent increase in our large sharks. So thinking about our observations versus our hypothesis, right? So our Mako sharks did in fact conform with our hypothesis. We're like, okay, that's great. Thresher sharks, they conformed with half of our hypothesis, if we can call that it, where we did see those high levels in the young, a decrease in juveniles, but instead of increasing as an adult, we see these leveling off of concentrations. And then finally, our blue sharks didn't conform at all. We didn't see any particular change. So kind of breaking those down then and thinking about what are some of the influences that might be resulting in why we're seeing these particular patterns. So first of all, focusing in on our maternal offloading, we did meet our hypothesis in the sense that the starting points of these animals are different for them by species. So if we break that down and we can compare those together, right? We had our Mako sharks that were the highest, followed by our Threshers, and then our blue sharks. So if we think about the physiology of these animals and what might be influencing some of those maternal offloading processes that would result in that, these three species use two different types of ways that they can provide supplemental nutrition to their offspring. So all of these animals are viviparous. That means they give live birth. Our Mako and our Thresher sharks use a similar form that's called oophagy, where essentially mom will ovulate eggs, unfertilized eggs, and those babies will eat those eggs for breakfast while she's pregnant in her uterus. Blue sharks, on the other hand, are placental. So they do have a maternal connection with the pups, but we think that this might be a reason why some of these animals are able to be more efficient at energy transfer that might influence how much contaminants can be offloaded. So if we break that down just a bit and thinking about these two forms of maternal reproductive investment, getting some rough estimates. Again, this is just based on number of pups and the weight of those pups. Again, rough estimates to kind of put some numbers on things. We see that our Makos have the highest investment in terms of the fact that they're creating the most biomass in a litter, followed by our Thresher sharks and then our blue sharks. So these differences in how they're able to provide that nutrition might influence, why we see those stepwise differences across the board in terms of the contaminants. But trophic position probably also has something to do with it too. And when we look at that, right, we have our Mako sharks that we are assigning them the first position. We are pretty confident that an adult female is going to be eating pretty high on the trophic level. But instead of having that be followed by a Thresher, it's followed by blue sharks, with Thresher sharks coming in third. So we think there's probably some interactions going on between our reproductive investment and our maternal trophic position. So coming back just for our final thing about this bioaccumulation problem, why aren't we conforming to those hypotheses that we set out? So if we focus on blue sharks first, what we think might be going on is that they have a slower metabolism of our three, which we think might be a lower feeding rate. They have more offshore lifestyle, so maybe that puts them further away from exposure because they're not spending as much time closer to shore, so closer to humans and where the contaminants are. And finally, they have that middle trophic position. So there might be some of this balancing that we're doing. And then finally, that low maternal offloading potential with that low investment might mean that, you know, they're accumulating, but they're also able to give off a little bit, and kind of the combination of all those things has a process whereby what they are taking in, they're matching either with maternal offloading or growth. And so that balance, oh God, someone's drawing on my screen. Anyway, so that balance might be why we didn't see those differential of accumulation like we were expecting. So finally, for our thresher sharks, right, we saw this decrease in the beginning, but a leveling off at the end. So what are some of the things that might be causing us to not see that? Right. So if we focus in on this latter part and think about the things that might be going on that could result in that pattern. So if we think about this again as a balance, right, between that intake and that growth or offloading, which is going to alter those contamination concentrations, thresher sharks have a higher metabolism, which we think might increase the feeding rate and therefore that intake. So that might skew it a little bit. But and they also have a more nearshore lifestyle that might allow them to have that higher exposure. However, they also are feeding lower on the trophic level, and they have a medium investment. So for our adults, it might be that those two things balance each other out, and that's why we don't see an increase in that concentration for our adult sharks. Finally, for our Mako sharks, again, these were conforming to our expectations. But if we focus in on the adults, there's some things that we can consider, and some data I'll show you that is going to hopefully back that up. So if we think again about this whole balance, right? So we're balancing that intake versus that output, if you will. So taking our large females, they have the highest metabolism of our three animals. So this increased feeding rate might tilt them towards having a higher contaminant input. Again, they have kind of this mixed where they're doing some things onshore and some things offshore. So maybe there's a higher exposure, hard to tell, but it would depend on if they're possibly feeding near shore, that could be an important thing. And then finally, that high trophic position, which is definitely going to increase the amount of contaminants that they're going to be accumulating. So they also had the largest investment, so the largest maternal offloading potential. However, what we think is going on is that despite the fact that they have that high potential, it probably isn't enough to completely balance out that scale like we saw for our thresher sharks, in the sense that they are going to accumulate those contaminants despite, in the face of these other factors. So if we focus on this, for example, and actually separate our animals from our older females, our younger females, and our adult males. So we have our males down here. This is the size at maturity. So we have we'll just call them younger females. So this is that subadult level. And then we have our definite adult females that would have for sure reproduced. So if we compare all those together and just extrapolate those curves, what we see is that the slope of these younger animals is actually much steeper than in our older animals, which might go to that hypothesis that yes, their maternal offloading is helping them to slow that accumulation rate, but it can't completely offset it based on the fact that they're feeding really high on the trophic level, and they have that high metabolism, which might mean that their general inputs are just going to be higher. And so these other processes can't balance that out. So finally, hopefully after this talk, I've demonstrated that we can use these other chemical tracers as tools that is an integration of ecology and physiology. So important to think about how each of those processes might influence both the signatures and the amount that these animals are going to accumulate. They do seem to be sensitive to species-specific ecology and physiology, which makes it really interesting then to go in and kind of put all those pieces together to see what we can learn about the biology of these animals. And finally, it does seem that the life history, so the specific characteristics of those animals are important when we think about things like accumulation and how much and what types of contaminants. So important to take these kind of holistic approaches when we're learning about the biology of these creatures. So with that, just a few acknowledgements to those that help collect tissues, as well as to Save Our Seas that actually funded this research. And if there's time for questions, I'd be happy to answer any of them. So at this point, I am going to close down the presentation so I can hopefully see some of your faces a little bit more.
Okay, thank you so much. Hang on just a second here. We'll get you set up. Okay, so thank you so much. Yeah, I have a couple questions just to start off with, and if anybody has questions throughout this, feel free to put them in the chat at any time. We'll come around to it. So we're going to open it up for questions and discussions. There was a lot of fascinating things to wrap my head around there. We've talked a lot in this series about bioaccumulation a little bit and just how that generally works. But one thing we haven't really touched on with contaminants coming up the food chain is just where you might find them. And I think with something like sharks, with having like such a wide range that they that they will swim through, and finding more contaminants in sharks that are closer to the shore versus sharks that are bigger or might accumulate generally more of the food web. Like, how did you kind of come across those findings really with, we would, kind of guess from just with those three sharks of finding like, well, we'd find generally more contaminants accumulating with the Mako shark because it's bigger and has a bigger range. But what about with ones closer to the shore, since you're mainly coming looking for contaminants that are coming from runoff, I'm guessing?
Yeah, so in Southern California, so it's both a runoff problem, but also a historic dumping. So Montrose Chemical Company, that was one of the major producers of DDT, actually disposed of their waste through the sanitation. So they just like literally dumped all of it in the ocean and then was like, oops. So we're still dealing with the effects of that today. But as to your question, so we can use some of those signatures to look at that coastal feeding. So our Thresher sharks, basically what we found is that despite the fact that they were eating the lowest on the food chain of our three sharks, they still had higher levels than a blue shark, which we know feeds higher on the food chain. And we attribute that to the fact that they're closer to shore. So they are just physically going to be eating prey that's interacting more with sediment that is associated with human activity. And therefore, just by that proximity, are going to have higher levels. And so other things we can look at are, I had mentioned earlier, a particular DDT signature in Southern California as like our kind of special thing. So actually, what data that wasn't shown here, but we can actually look at the change of that DDT signature over the lifespan of an animal. And in Threshers, it pretty much stays the same. And we would expect that because they are coastally associated species. They're not making these onshore, offshore migrations. And we see a lower signature for our blue sharks, which are feeding more offshore. So it definitely is this balance between ecology, but also like physiology of the animals. So sometimes it's hard to parse those apart, but essentially the main take home point to answer your question is that we have to consider kind of all those factors. It's not just the biggest animals, but it's where you're feeding on and what you're feeding on, that the combination of both of that's going to be important to how much you accumulate.
It's also pretty, it's interesting to to hear about DDT, because previously, for those of us who have joined in on these talks previously, we've heard a few researchers talking about bioaccumulation, and we're usually talking about mercury, nitrogen, things that are just coming through more, I would say, like contemporary pollutants. DDT is a bit of a throwback, and it's wild to think about that still being found in current shark populations. So when you were talking a little bit earlier, you know, a big part of your research is studying how that passes through from generations of sharks. So with that DDT, first of all, how long does DDT really exist in the environment like that? And is it mainly being passed from shark to shark through generations, or is there still DDT in the general ecosystem off those waters?
Yeah, great question. So I would say it's probably both. So DDT, if I remember correctly off the top of my head, has a half-life of about 50 years. So half-life basically means that that if you took the parent compound of DDT, and there's actually two different types of DDT based on the chlorine positioning, it'll take 50 years to go from that parent compound to a daughter compound, but it's still a metabolite of DDT. So it doesn't mean that it goes away. It just shifts from going from the parent to a daughter. And there's actually several daughters down from that are still in the environment. So we can look at the proportions of those different types of DDTs to make inferences on the degree of exposure from a particular area. So for example, some places in Africa still manufacture and use DDT because it's such a good pesticide for controlling vector disease, like other diseases that we're worried about through things like mosquitoes. And so if you looked at animals over there, you'd actually see a different ratio of that DDT that indicates a more recent use. So essentially this stuff stays in the environment a really long time. DDT was banned from production in the US in the 1970s. So, you know, that was like 50 years ago, and it's still a problem. And I think that it definitely shows that just the lasting effects that we as humans can have on an environment for chemical production, which has not slowed down. So even though it's a kind of this distant memory, it's still clearly an issue. So what we see though is that, I mean, in Southern California, there will be warnings on piers to not eat fish because the sediment is still pretty contaminated. So there are cleanups and there are remediation efforts, but it's just very difficult for the literal tonnage of hazmat waste that is in the environment to clean up. So it is passed generationally, but they also are going to acquire it from their environment too.
Wow, and that's and you know, as you mentioned, DDT was stopped being manufactured in the United States 50 years ago. You say it's still in wide use in other parts of the world. And the ocean covers a lot of that. So that just has me thinking, are there other contaminants, including DDT, I guess, that you would see in shark populations, say off the coast of Southern California, that might be coming from somewhere else that, you know, might not be used in Southern California, but would it disperse enough over the currents to not affect those shark populations, or maybe the shark populations there interacting with other shark populations in other parts of the water? So how important, I guess, what I'm getting at is a more global effort to reduce contaminants that are getting into ecosystems?
Yeah, I mean, great question. So I think we have to take a bit of a wider lens with that. So in particular, there are some pesticides that previous use in Asian countries, actually, you can see a coastal wide, like along the West Coast. And that's because based on atmospheric patterns, so when these things are released into the environment, into the air, how air moves across the Pacific, it forces those contaminants actually to the poles. So you'll get a trappage of contaminants poleward. And so there are some specific pesticides that were continually used in Asia. I'm not sure if they're used currently, but you would see like animals in Alaska that would have a particular signature that would differentiate itself from something that's in Southern California, just based on how the air moved across Asia up to the polar ice caps, basically. So it's why like very pristine environments like the Arctic have problems with contaminants, even though we don't manufacture contaminants, you know, in icy places. But that atmospheric deposition is affecting polar bear populations in terms of some of their reproductive parameters. You know, climate change and no ice aside, they also kind of are battling human influences on two fronts. So I think that doesn't like explicitly answer your question in terms of like, can you point to like an example of like X contaminant that that would have come from somewhere else? But I think the point being that yes, we're all connected, and understanding, you know, physical processes of, you know, oceanography, hydrology, and kind of, you know, not just focusing on just biology is going to have an influence for these sorts of things and the animals that we care about.
Yeah, absolutely. And again, like essentially what we're talking about is these contaminants are coming from their use on land, right, and running off into the water. And it's that can come from so many different ways, and whether it's something like DDT being used in agriculture, you know, in the Salina Valley 60 years ago, and making its way off the coast of Southern California now, or we have a very interesting thing that happens here in South Florida, usually in the late spring, early summer. We'll have, if there's a big dust storm in the Sahara Desert, it'll make its way over here, and we'll have dust in the sky. The sky kind of turns this orangish thing, and so that dust is, you can picture it also raining into the ocean, basically in between here and there. So anything that might be in that dust is getting into that ocean also. And that's part of the natural process, and there's minerals that are important to that. But if there's things that might be harmful, that's in there too. So the interconnectedness is and so I think the way that you're studying that is very fascinating, and tracing those specific molecules and how they exist in those populations is something that's really, yeah, just something, I mean, I haven't thought about before. And, you know, we've been talking to, we've heard from a number of different researchers, and this is the first time we've heard anything quite like that. So I'd kind of like to know, like, what led you to like that? Like, what led you to that field of research? I think one interesting thing about doing this is just hearing, like, how did you get into doing what you're doing? Because that's a pretty, you used the word niche a few times. And like, that's a pretty niche.
Yeah, like how did you arrive there? Yeah, so my general background is interest in maternal offloading processes. So I did my master's examining in a model elasmobranch species around stingray to look at how that transfer actually occurs, and how that varies by sex. And kind of that just branched off from there of like, okay, well, what if we look at other elasmobranchs that reproduce in different ways? You know, will a specific type of strategy produce more offloading to young than others? So as you might imagine, it's kind of difficult to get your hands on pregnant, you know, Mako sharks that are meters long, that are big animals, and that sort of thing. But we actually had an opportunity to sample a pregnant Thresher shark, and we were estimating that just based on what we knew in the pups that she had and what she had in her and what mom had in her liver, that mom's capability of offloading was pretty high. It was between 30 and 50% of her contaminant load. It seemed that she was able to pass to her offspring. And again, that's using that very specific form of matotrophy, that oophagy, where mom is making these really yolky eggs, so full of fat, full of nutrients, but also is going to be full of contaminants that are coming from the liver, because those contaminants just bind to fat and they just follow wherever the fat goes. And so that's kind of how this spurred some of this, is like, oh, well, we should look at contaminant concentrations in these different sharks that, you know, share a commonality. So we had a different paper that looked at Thresher, Mako, and white shark across juveniles, to try to get at that. So they all shared a common reproductive mode. And then we just expanded that and said, okay, well, let's try some different ecologies, and what are some of all the factors that are going to influence. So that was kind of the starting point. And then it just branched into like, oh, like we can distinguish between species. This is kind of crazy that we'd be able to do that. And it just kind of led to more questions than answers.
Yeah, it's right. I mean, it's funny to see.
Yeah.
Yeah, exactly. It's funny to see the parallel of just like what you study and how you arrive at it, kind of doing the same thing of just like uncovering one little thing and then just watching the chain reaction kind of go up and magnify. Really fascinating stuff. I know we only have a couple minutes left, so I just kind of want to ask, you know, is there any just a simple way for our audience to be aware of or get involved in any issues that have come up in your research that you know, that would draw down, whether it's knowledge of seafood that they're eating, swimming habits, just what to really be aware of?
Sure. And I mean, I'll take, I guess, putting my toxicology hat on. Mm-hmm. I would encourage people to think about the types of products that they use in their everyday household. So sure, like you can't, you know, the DDT problem isn't because like a lot of people went to the store and bought DDT and then sprayed their lawns. That being said, there are other contaminants that are currently a big problem, and we're only starting to learn about, that we're like, oh, yikes. So I would encourage people to think about, you know, the types of products that they use in their everyday household, and maybe take an introspective thought about like, well, where does this come from, and where is this going, and can it be removed in the wastewater treatment plant? You know, if it's something that you're flushing down the toilet or applying to your skin, or, you know, any of those things of like, is this going to result in the ocean? And can I find some alternative that might be something friendlier to use? So from a toxicology standpoint, I would encourage people to think about that. But also about, I mean, it comes down to regulations, right? And supporting efforts of making sure that we're keeping companies accountable for what they do and how they do that. So just kind of considering those variety of aspects, I think would be a good place to start. Again, keeping my toxicology hat on.
Absolutely, and thank you for that advice. Again, like, you know, one thing to keep in mind, everything we're doing is connected to one other thing and goes up from there, and nothing really goes away. So
unfortunately.
Right. So, well, thank you again. Please join me in thanking us, Dr. Lyons, for presenting tonight. It was a wonderful presentation, and good luck with everything at the Georgia Aquarium and with future research. And we hope to hear from you again soon.
Thank you so much. It was a delight.
All right. And so we're going to wrap it up now. First, I want to once again thank Dr. Lyons for joining us, and thank the Save Our Seas Foundation for making this possible. We have a couple more virtual Distinguished Speaker Series events coming up. Next one being on August 5th. We'll be talking about microplastics with Alina Werserzek, a PhD student at the University of Ireland, Galway, maybe the furthest away
that we've talked about. She'll be joining us on August 5th, and then we'll be having a manta ray panel discussion with Dr. Guy Stevens and Emily Humble from the Manta Ray Trust on September 2nd. Those will both be virtual events, same as this, 6 p.m. the first Thursday of the month. We also have a wide range of things happening down here on site at the museum all summer, so please come down and visit us. And thank you again for joining us tonight. I'm Brady Newbill signing off. And we'll see you again next time. Good night.